Okay, good. Welcome, everyone. In this talk, we're going to be going over the most predominant current diseases in barley from the east to the Midwest to the West. I'm very happy and honored to have been to follow this talk up by the great Dr. Brian Stephenson, who gave a wonderful overview of diseases he's been working on. So I won't touch so much on the stem rust, the leaf rusts, but we'll be going over some diseases that you are aware of that he has just mentioned prior to this talk. And we'll be focusing on new avenues of barley growth and development, including the winter barleys, what unique aspects that they have to do and encounter, and what new unique diseases barley growers are dealing with from around the country in different regions with different climatic issues. I'm Thomas Baldwin, Dr. Thomas Baldwin from North Dakota State University. I'm the barley pathologist there, assistant professor of barley pathology, and we deal with barley diseases and study them for advancements of germplasm, developing products to stave barley disease, and to learn about them. So myself and my graduate student, Joan, here are looking at barley plants, and we're probably seeing a plant that might look like this, which has specific striping patterns we believe might be due to barley stripe mosaic virus or a similar pathogen causing this disease. And if you zoom in a little bit, you can see that something's not right with this barley. There's a unique aspect going on causing a symptomatic pattern that we have to go and identify. Now, the question is what are barley diseases and what is barley disease? For most farmers, it's something's not right with my barley. Something is wrong with the way my barley looks in the field, and what's going on and how do I fix that? Talking to Suzette Baldwin, who's the plant diagnostic lab manager at North Dakota State University. Most of the time when she gets these issues, we're looking at biotic stresses, where something is off in the field, low nitrogen or other nutrients, and they resemble a pattern that could look like barley disease. But some of the times she gets in samples that look like this, the biotic stresses, where particular patterns of disease, whether it be leaf lesions or symptomatic coloring, like we just saw on the last slide, are being caused by an organism which is causing and manifesting those symptoms. And we want to obviously treat those differently. So completing our wheel here of things that we don't like are some of the diversity in barley disease that you can see in the field. And we'll just keep this nice wheel here to help us guide forward for our presentation. Now, these barley diseases can be caused by many different types of organisms, but in general, of course, we know bacteria, fungi, viruses, and nematodes can all be responsible for causing barley disease, and we want to characterize each of those. So the question is for the pathologist, the extension specialists, the barley breeders here, they understand why barley disease is a problem and why you should care about them. But how about the other members of AMBA, your maltsters and brewery industry folks? Why would you care about barley disease? And the answer is majorly, of course, economic losses caused by barley disease. Yield loss, of course, we all know of losses due to, say, Fusarium head blight with loss in quality, but even diseases, foliar diseases, can cause structural decay of malt and also cause many issues to reduce that yield loss, causing increased cost of testing, cost in time to have everyone handle and measure those. So it's a huge time waste. It's a waste of product if you have to throw that out. And you have inputs like fungicides that you have to deal with in order to do it. So So, but we have malt quality concerns. Of course, everybody knows Don, but there's also structural deterioration that could be caused with increased disease pressure, mycotoxins, and reduced brewing performance, specifically lautering. So this is a systematic effect that will happen from both the farmer up to the maltsters, the brewers, and even down to the consumers, as we've seen with Brian Stephenson's slide, that the beer could be costing more and consumers aren't going to like that. Just focusing on some of the economic inputs, Dr. Andrew Friskob has determined that, in a sense, foliar disease, despite having resistant lines, despite using foliar fungicides, is costing North Dakota barley farmers an estimated $1 million in yield loss each year. And then just to highlight how the absolute worst it can get, in North Dakota in 2013, it was estimated an annual loss of $250 million and 40 million bushels due to the contamination on Fusarium head blight. This is an important statistic. I always bring it up because it's the high point, but is actually, we've lost more in 2013 than we currently produce today in North Dakota. So that gives you an overview of how important these diseases are and can be. Of course, our major problem that we were talking about with malt is Fusarium head blight, with Don and also the hydrophobins contributing to the beer challenge that we know of as gushing, where this makes a product that is inferior for the market, and you can't have that. So we don't want that in our beer. Now, when we think about barley disease, just to break it down, the easiest way is to think of it by timing, when we're from planting to harvest, to so by stage and by region of the plant itself. So starting at the roots, the crown, moving up into the foliar where the leaves and the stems are, and then up into the spike region. So just by region, if we take a look at the roots, we're dealing with problems such as common rots, Fusarium crown, and root and crown rots. And both of these we rarely see in our eastern half of the state, North Dakota, but far out west in western North Dakota and the beginning of in the eastern part of Montana, where it's nice and dry. These are conditions that kind of favor these roots that we see in the field. These are stand reducers, so they reduce your overall yield from the crop that year by killing off the plant early, affecting their root system. These, of course, are being caused by fungal diseases, but we have other issues such as cereal cyst nematodes, which are also a problem, mostly in sandy soils in places like Idaho and Montana. And then moving up into our foliar region, we have important foliar diseases such as scald, net blotch, which we have two forms of net blotch, which are net form net blotch and spot form net blotch. And these can actually be confusing and difficult to pick out in the field. Our other important foliar disease, which is spot form net blotch, a good way to tell is with the yellow corona that you see around the spots in the spot blotch. But to really be sure, you want to take that and bring it into the laboratory itself and look under the microscope and see what the fungus looks like. And looking at this fungus in here, you can see the cells, which are pill-shaped, elongated, and transparent type cells, which is Pipharena teres, form Specios teres, the net form net blotch version, opposed to the spot blotch, which is Cochleobolus sativa. And you can see the dark Hylian double-celled, which is why they used to call it bipolaris. And these are very distinguishable in the laboratory, but not so much in the field, and they can actually co-occur in the field, causing a disease complex. So that's where we are in barley disease. I would be amiss if I couldn't at least mention a little bit of rust. So in our foliar disease, our most important rust is stripe rust, and we will see this on occasion in the field. One of the newest members, a non-fungal pathogen, a bacterial pathogen, is actually bacterial leaf streak. And with the addition of the Barley Pest Initiative funds, which we could not have been possible without AMBA's help, we've been able to do extensive research in this emerging pathogen, which you're about to find out is an important pathogen in the field and is emerging as an important pathogen. I just want to introduce my team of Dr. Zauhi Liu, Jel Velasco, and now Glenn Rose Belen, our main researchers working on bacterial leaf streak that we have some exciting things to show you about. But overall, the main different thing that we're getting across in foliar disease is that you have a field and that field requires the photosynthetic capacity in the leaves in order to produce and fill the grain. And if your leaves start to look like this because they're being attacked by foliar pathogens, then you're losing out on that yield potential. Moving on to what most people are aware of or identify as like the diseases that they are most aware of is spikes. So the spike diseases that are primarily important, we have Fusarium head blight, of course, with the brown water soaking lesions, ergot and loose smut being the most common ones that we see. Fusarium head blight being the number one issue. You can see it growing here on this fluffy culture on the left, and this pinkish hue. And it gets so bad in the fields that we can actually see parathecia actually developing right on the heads themselves. My graduate student Avea Navaska has been working with this pathogen along with other fungal species and has developed a qPCR test in order to quantify the levels of Fusarium that are actually in both the grain and the malt. And that's available through a couple of publications. So where are diseases and where are they most important? So as we say, the climatic regions throughout the United States is very diverse. So I wanted to ask the experts that are in these regions what they're seeing in the field for barley disease and what we're doing against it. So as we can see, the most predominant states being Idaho, North Dakota, and Montana. And these are the general regions, but then we have what I like to say re-emerging regions of barley cultivation, which is including the East, Virginia, New York, and say South Dakota. In the Midwest, talking to our cereal extension specialist, Dr. Andrew Friskoff, I asked him what his feeling on barley disease is in North Dakota. And his thoughts are Fusarium head blight is still number one. It's at a static level where it's a continued nuisance, but it is being managed. Foliar diseases are a continued threat, but the fungicides and the host resistance that we have in place are keeping up. It was a major problem back a decade ago, and now is curtailed into something that we can manage. And overall, he's concerned about BLS, bacterial leaf streak, becoming a problem and being a problem with no management solutions yet on the table. Moving on to Idaho, obviously an important region. I talked to Dr. Juliet Marshall, who is Department Head of Plant Sciences and Professor of Plant Pathology, what she thought about barley disease in her region, and specifically on winters, because North Dakota doesn't have winters and Idaho does. Disease affecting winters and spring barley, she says, are largely similar, although there are some observable exceptions, including cereal cyst nematodes, which is actually an advantage towards winter barley. Bacterial streak poses a significant problem last year and is likely worsened by environmental factors. So I'm seeing twice we're hearing that bacterial leaf streak is an issue. And Scab was an exceptionally aggressive pathogen on 1 year. Moving out eastward, talking to Dr. Gary Bergstrom at Cornell, with barley being an emerging industry in New York, I asked how things are going, where they're primarily growing winter barleys. In his experience, winter malting barley, although many diseases have been recorded, only a few had impacted production, mainly FHB. And if he was to rate them, he would say that malting barley for winters, the problems would be first, Fusarium head blight, second, scald, third, leaf rust, and then fourth, other leaf blotch complexes. Baldwin is an important foliar disease in winter barley. This is the second time now I'm hearing that scald is an issue. So what are we doing about this? We are doing a lot of breeding and screening for these diseases here in North Dakota, where I manage both Fargo and Langdon disease nurseries. We put about 8,000 plots in the field last year, looking at spot blotch resistance and FHB resistance. Here's my field specialist, my field specialist, Abraham Hayamashel, not doctor yet, doing some field screening. This takes a lot of time and a lot of effort, but it continues our germplasm to be at least to the levels where we expect it at for being head blight resistant. Of course, this takes a lot of effort, but it is completely necessary in order for us to maintain the resistance that we see in our germplasm. But what we want to do is we want to move forward, and how are we going to do that? I see a couple of technologies on the horizon. First off being gene silencing technology, in which we have a gene that comes from Fusarium itself that is able to shut off specific genes in Fusarium. I think this has great potential, but we need to learn a lot about how the plant can produce this gene, how the fungus will pick this up and silence its own genes. A next trait that I see as a potential technology for moving forward is something known as spore killer trait, which is a natural gene drive which will knock out spores in the asci that don't carry this trait. It's like a self-killing type thing, lowering the population and also editing out things that we don't want, such as deoxynivalenol. And then the third technology I see coming along is the microbiome defense, which we were just getting into and just understanding and touching this iceberg of potential. Is that, as we know, Fusarium is not the only species that's involved in barley growing in the environment. There's lots of other microbes that are out there. And when Fusarium is actually in the mix through our recent evidence, we can show that the microbes are reacting to Fusarium being there and shifting dramatically. And in theory, actually, some of them will be helping to prevent FHB from producing, helping to prevent FHB, and some of them might be helping FHB to cause disease. And we want to weed those out and get rid of them and reduce the amount of FHB in the field. This is being led by a team of scientists such as myself, Dr. Brianna Whitaker, and Dr. Barney Geddes at NDSU, along with my grad students, Jonah Casso and Brooke Benz. And finally, what are we doing for bacterial leaf streak? Bacterial leaf streak, as we see in 2 states, have been identified as real problematic. And what are we doing? Dr. Andrew Friskop is looking at management strategies, spraying a lot of things. The bad news is he's tried everything, including the kitchen sink, and has not been able to find management strategies that are effective against this, but we're still trying new formulations in order to get new results. We just got done doing whole genome surveys on isolates in Idaho, Montana, and North Dakota, thanks to support from AMBA and funding from the Barley Pest Initiative. And we're going to come out with that paper soon. And what we found is there's a dramatic amount of diversity in the pathogen population in terms of the pathogenicity genes that they have in them to cause barley disease. And we're seeing how that affects the overall pathology. And then the third thing we're doing is screening for resistance. My graduate student, Jel Velasco, has spent a lot of time and effort developing screening protocols, and we believe that we have found actually true resistance to bacterial leaf streak in some barley lines and are able to share that. Because this is such a big issue, I would like to invite people here, April 1st to the 2nd, go to blsresearch.org. We are actually holding a meeting to address bacterial leaf streak research in both wheat and barley. Summary slide. So just to go over what the future of barley disease entails and what we want to do. We want to move the needle forward on FHB. We want the next 10 years not to look like the past 10 years. We're actually making some decent progress, And a couple of those tools that I highlighted might be some things that we're using to get that job done, along with advanced breeding techniques and increased screening. We want to be proactive on bacterial leaf streak. We don't want bacterial leaf streak to be Fusarium head blight in 1996. We want to get ahead of it. We see that there's a massive amount of diversity and management that needs to be done. We want to identify resistance already and have that into the germplasm. And third, we want to know what's next and what's going on. In two of those slides, talking to two of my colleagues, scald has become an issue. And this is taking me aback because we don't see scald in North Dakota. So we really want to maybe have that on our radar and see what we need to do moving forward. Do we need to start working back on scald and start finding solutions for that management issue? I'd like to thank you all for listening, and I'd like to thank, of course, the U.S. Wheat and Barley Scab Initiative for their continued support on our projects, the North Dakota Barley Council, and also, of course, American Malting Barley Association for helping us fund all of the screening that we do, the Barley Pest Initiative work, and being supportive on our research staff. Thank you very much. Ming. Oh, yes, go ahead.
Are you able to say what are your sources of unsafe fugitive resistance for that BLS?
Yes, we have found a line that we can't find the source for anywhere, and we're currently we're characterizing it, doing some mapping population and sequencing it. But yes, I can definitively say I would give you the source. We have no idea. So the background on that is Gel, my grad student, was running massive screening programs and found a seed lot that was marked Pinnacle because that's what we were using as a check. And this seed lot happened to have come from one of our incredibly diverse old FHB field trials just on the border. And he found one line that was producing a hypersensitive response to bacterial leaf streak. And we just cultivated it, moved it up from there. We went to genotype it, and there was nothing like it in our genotype banks. So kind of fell out of the sky. But we are certainly willing to share that germplasm. And we're also working with Dr. Stephenson and the lines that he's picked up looking at field trials. Dr. Yang?
You're also my question.
Okay. Dr. Whitaker. Right. Yeah, so the screening technique that we've developed, we do a combination of spray inoculation. This is how we found that line, by the way. So we have spray inoculation, which we can give you the details on. The main, I guess, secret sauce that we've developed seems to be cycling the humidity in the greenhouse that allows us to get really sufficient infection and good screening.
How many different isolates do you use?
We're using, okay, so we are just using BAF1, which is our North Dakota isolate, but we're screening now a panel of barleys, about 20 barleys, to look at at least representatives from each of those three clades. We have about 200 isolates that we have full genome sequencing on that we've characterized, and the diversity, like I said, the diversity and talon effectors, even type secretion systems are pretty astounding. Yep, Nicholas, see you in the back.
Tried any like in-field inoculation stuff?
Yes. For BLS? For BLS, yes, we do in-field inoculation trials, mostly with Dr. Andrew Friskop and Dr. Zhahui Liu. We use basically, the backpack leaf blowers with a combination of our one isolate that we put in the field and a solution to get it on. I forget the exact formulation. And it does, it's actually quite extraordinary. I think it's fine for me to talk on this with Dr. Friskop, is that it's a leaf blower and he'll blow down a row and on either side he'll have checks and he doesn't see disease on those checks, probably because the leaf blower is putting wounds in and allowing that pathogen to entry. What I've actually learned from the microbiome work is that Xanthomonas, and specifically Xanthomonas translucens, is pretty ubiquitous in the environment. So understanding what triggers that bacteria to produce disease or just stay endophytically is going to be a hot topic in research, I think.
I haven't seen
it. Okay. I wouldn't. We can test your stuff for you. So, thank you.